For as long as humans have been studying the natural world, we have tried to organize it. We like labels. Mammal, reptile, fish, insect. We build neat little boxes and then we expect everything we discover to fit cleanly inside one of them. But every once in a while, something is found that completely ruins the system. A creature that looks like it should not exist. One that mixes traits from totally different branches of life. Something so confusing that scientists argue about it for decades. Sometimes over a century. At first glance, it looked like something from a nightmare. In 1958, an amateur fossil collector named Francis Tully was searching for fossils when he came across something so strange that even experienced paleontologists were baffled. The fossil had a soft, torpedo-shaped body. It had a long, flexible proboscis extending from its head, ending in a claw-like pincer. And on top of its head, it had two stalked eyes that stuck out sideways like a hammerhead shark. This creature became known as Tully Monster gregarium, or the Tully Monster. The problem was simple. No one could agree on what it was. For decades, scientists debated whether it was a worm, a mollusk, a chordate, or something entirely unknown. It did not match any known group of animals. It had features that seemed to belong to completely different evolutionary branches. Some researchers argued it was related to the lamprey. Others thought it might be an invertebrate. At one point, it was even suggested that it belonged to a totally extinct branch of life with no modern relatives. The fossil record did not help much either. Tully monsters were found only in one specific geological layer from about 300 million years ago. They did not appear before that period, and they vanished completely afterward. Even modern imaging techniques caused more confusion. In 2016, a study suggested the creature might have had a notochord, which would place it among vertebrates. But later studies changed that interpretation, arguing that structures were misidentified. As of today, there is still no universal agreement on where the Tully monster belongs. It is the official state fossil of Illinois, and it remains one of the most stubborn classification problems in paleontology. When European scientists first saw it, they thought it was a joke. In 1798, a preserved specimen of a platypus was sent from Australia to England. It had the bill of a duck, the fur of an otter, the tail of a beaver, and webbed feet. Naturalists genuinely believed someone had stitched together different animals. They checked for seams, they tried to peel the bill off the body, assuming it was glued on. The platypus completely broke the biological rules of the time. It was a mammal, but it laid eggs. It had fur, but also a venomous spur on its hind legs. It produced milk, but had no nipples. There was no category for it. For years, scientists debated whether it should even be considered a mammal at all. It did not fit the definition as they understood it in the 18th and early 19th centuries. Eventually, researchers realized the platypus belonged to a very ancient group of mammals called monotremes. These animals split off early in mammalian evolution and retained reptile-like traits such as laying eggs. But at the time of its discovery, that category did not exist. The platypus forced scientists to rethink how mammals evolved. Even today, it remains one of the most biologically strange animals alive. Its genome is a mosaic of reptile, bird, and mammal genes. It is not an evolutionary mistake. It is a survivor from a very old chapter of life on Earth. When it was first reconstructed, it was upside down. Hallucigenia was discovered in the Burgess Shale fossil deposits in Canada, which date back over 500 million years. These fossils preserve soft-bodied creatures from the Cambrian explosion, a period when animal life rapidly diversified. Hallucigenia looked like a thin worm with long spikes sticking out of its back and soft tentacles underneath, or at least that is what scientists thought. Early reconstructions placed the spikes on the bottom and the tentacles on top. This made the animal look like it walked on sharp stilts while waving soft appendages in the air. It was so strange that the creature was named Hallucigenia because it looked like something seen in a hallucination. Later discoveries revealed the entire reconstruction was wrong. The spikes were actually defensive structures on its back, which to be fair, makes a lot more sense. The soft tentacles were its legs. Even the head was misidentified. Eventually it was linked to modern velvet worms, but it represented an early and very different body plan. It was part of an extinct experimentation phase in evolution where nature was trying out designs that would not last. Hallucigenia showed scientists that early animal life was far more diverse and bizarre than previously imagined. It forced a rewrite of how complex animals evolved. In 2004, scientists working in the Canadian Arctic uncovered a fossil that filled a massive evolutionary gap. The creature was named Tiktaalik, and it lived about 375 million years ago. This creature had scales, fins, and gills like a fish, but it also had a flat head, lungs, a rib cage, and wrist bones capable of supporting its weight. Most importantly, it had a movable neck. Fish do not have necks. This creature did not fit cleanly into fish or amphibian categories. It was something in between. Before, scientists knew that vertebrates moved from water to land, but the transition was poorly understood. This creature showed how that happened step by step. Its fins were structured like primitive arms. It could prop itself up in shallow water. It likely spent time both swimming and crawling. At the time of its discovery, it did not fit any existing classification. It forced scientists to accept that major animal groups did not appear suddenly. They emerged gradually through transitional forms. For decades, no one could agree if it was even an animal. Dickinsonia fossils are oval-shaped, ribbed impressions found in rocks over 550 million years old. They come from the Ediacaran period before the Cambrian explosion. The creature looked like a flattened pancake with symmetrical segments radiating from a central line. Scientists argued endlessly about what it was. Some thought it was a jellyfish. Others said it was a fungus. Some believed it was a colony of microbes. Others claimed it was not an animal at all. The problem was that this creature lacked clear features like a mouth, gut, or limbs. In 2018, researchers discovered traces of cholesterol molecules preserved in the fossil. Cholesterol is associated with animal cells. That finally confirmed this was an animal, but it still did not fit into any known animal group. It represented a completely extinct lineage that did not survive into modern times. This showed that early animals followed evolutionary paths that later disappeared entirely. For a long time, it was dismissed as a myth. Local people in the Congo rainforest spoke of a strange animal called the forest giraffe. European explorers thought it was folklore. In the early 1900s, physical evidence finally emerged. The Okapi looked like a horse-sized animal with zebra-like stripes on its legs, but it had a long neck and a skull structure similar to a giraffe. When scientists examined it, they realized it was the giraffe's closest living relative. The problem was that it looked nothing like one. It lived in dense forests instead of open savannas. It had adapted to a completely different environment. Before its discovery, no one suspected giraffes had forest-dwelling relatives. The Okapi forced scientists to reconsider giraffe evolution and how flexible large mammals could be. For over a century, scientists argued about its teeth. Helicoprion was a prehistoric shark-like fish that lived over 270 million years ago. Its most famous feature was a spiral of teeth arranged like a circular saw. The problem was that no one knew where the spiral was located. Some thought it was on the nose, others thought it was on the tail, some placed it inside the throat. There was no modern equivalent. Only with modern CT scanning did researchers finally understand the structure. The spiral was part of the lower jaw, growing continuously and curling inward as new teeth formed. Helicoprion did not fit into known shark groups. It belonged to a now extinct lineage of cartilaginous fish with bizarre feeding mechanisms. Its discovery showed that ancient oceans were filled with predators far stranger than anything alive today. It was supposed to be extinct. Coelacanths were known only from fossils dated to over 66 million years ago. They were considered a classic example of an evolutionary dead end. Then, in 1938, a living coelacanth was caught off the coast of South Africa. Scientists were stunned. The fish had fleshy, limb-like fins similar to those of early land vertebrates. It had a unique jointed skull and an oil-filled organ instead of a swim bladder. It did not fit modern fish categories. The coelacanth was a living fossil, but not a primitive one. It was highly specialized and well adapted to deep sea water environments. Its survival forced scientists to rethink extinction assumptions and evolutionary stasis. At just over 3 feet tall, it shocked the world. In 2003, researchers discovered skeletal remains of a small human-like species on the Indonesian island of Flores. The species was named Homo floresiensis, often called the Hobbit. It had a small brain but sophisticated stone tools. It lived surprisingly recently, possibly as late as 50,000 years ago. Scientists argued whether it was a diseased modern human or a separate species. Further analysis showed it had unique skeletal traits not seen in Homo sapiens. It did not fit existing human categories. It represented a separate branch of the human family tree. Its discovery showed that multiple human species coexisted far more recently than once believed. Its face looks like a creature from science fiction. The star-nosed mole has 22 fleshy tentacles around its nose, each packed with sensory receptors. It is the fastest-eating mammal known, capable of identifying and consuming prey in milliseconds. Its nose functions as a touch-based sensory organ, unlike anything else in mammals. When it was first studied, scientists struggled to classify its sensory system. It showed that mammals could evolve sensory adaptations far beyond traditional sight and smell. Alright guys, that has been our list for today. Thanks so much for checking it out. I've been your host today, Olivia Kozlowski, and I will see you again soon. Goodbye.